There's a deceptively simple question at the heart of P10 neuroscience. When you knock out P10 in a neuron, the PI3 kinase-AKT-mTOR pathway goes haywire. But which node on that pathway is actually responsible for the cellular pathology? mTOR gets a lot of attention, but AKT sits upstream of both mTOR complexes and has dozens of its own substrates. Whether AKT itself was the necessary driver versus just a conduit to mTOR had never been definitively established in neurons. That's exactly the question Brian Lucart's lab at the University of Alabama at Birmingham, in close collaboration with Matt Weston's group at Virginia Tech, has now answered. Their paper, AKT is necessary for neuronal hypertrophy and epilepsy caused by Pten knockout, places AKT squarely at the center of Pten loss pathology, not mTOR downstream. AKT.
To understand why that distinction matters, it helps to know what Pten loss actually does to a dentate gyrus granule neuron. The cell becomes dramatically enlarged. Soma area roughly doubles. It sprouts an over-elaborate dendritic arbor with increased primary dendrites and total dendritic length. Its spine density doubles, driving a greater than twofold increase in excitatory synaptic input. It migrates aberrantly into the wrong position within the granule cell layer. And collectively, these changes produce hyperexcitability that, when enough neurons are affected, tips into frank epilepsy.
Prior work from the Lucart and Weston labs had established that mTOR C1, downstream of AKT, is a major driver. Knocking out raptor rescues hypertrophy, migration, and synaptogenesis. Knocking out Richter, which assembles mTOR C2, partially rescues some phenotypes but not others. The picture was complex, and it was unclear whether AKT itself was necessary or whether other PI3 kinase effectors could substitute.
The approach Prina, Goyette, and colleagues used was elegantly intersectional. Using retroviral delivery into neonatal mouse dentate gyrus, they knocked out P10 along with AKT1, AKT3, or both simultaneously, in individual neurons side by side with wild-type controls in the same animal. They could also overexpress human AKT1 or AKT3 to reconstitute signaling in the knockout background, giving them a complete gain and loss of function toolkit.
The headline result was unambiguous. Knocking out either AKT isoform alone in the P10 knockout background rescued very little. The two isoforms compensate for each other nearly completely. But the triple knockout, P10, AKT1, and AKT3 all gone, was transformative. Soma hypertrophy was almost fully rescued. Dendritic arborization and primary dendrite number returned to wild-type levels, spine density normalized, and excitatory synaptic frequency measured by spontaneous EPSC recordings was indistinguishable from control neurons.
One finding deserves particular attention. In the triple knockout, neuronal migration didn't just return to wild-type, it actually dropped below wild-type levels. P10 knockout neurons migrate too far into the granule cell layer. The triple knockout neurons migrated less than normal neurons do. That over-rescue is telling. It suggests AKT is regulating migration through mechanisms that extend beyond mTOR signaling, possibly through substrates like PALD1 or GERDin that haven't yet been fully characterized in this context.
The electrophysiology was the functional confirmation. Whole-cell patch clamp recordings from triple knockout granule neurons showed membrane capacitance and spontaneous SEP-SC frequency that were statistically indistinguishable from wild type. These cells, which would have been hyperexcitable due to P10 loss, were completely normalized by removing AKT.
The most clinically striking result came from the POMC-CRE model, where P10 is knocked out in a substantial fraction of developing granule neurons. These animals develop severe temporal lobe epilepsy, dentate gyrus foliation, ectopic basal dendrites projecting into the hilus, spontaneous seizures, and die prematurely around postnatal day 80. It is a compelling model of P10-related epilepsy in a genetically defined population.
The triple knockout of P10, AKT1, and AKT3 in P, OMC, Cre-expressing neurons rescued all of it. Normal dentate architecture, no ectopic dendrites, and complete restoration of lifespan. This is a significant proof of concept that the Pten-AKT signaling axis is the critical link between cellular dysregulation and overt epilepsy, and that targeting AKT is sufficient to prevent the disease.
There is also an important isoform story here. AKT3 is the dominant isoform in hippocampal neurons, comprising roughly 2/3 of total AKT in the dentate gyrus. AKT3 overexpression drove greater increases in soma area and distal dendritic arborization than AKT1 overexpression in otherwise identical conditions. The differential contributions of these isoforms to specific morphological compartments are not yet fully explained, but the patterns suggest they may be engaging at least partially distinct downstream programs.
Which brings us to the most intellectually provocative implication of this paper. Establishing that AKT is necessary is a major finding, but necessity is not the same as mechanism. We know AKT activates mTOR 1 through S6 kinase and drives a cap-dependent translation and growth program. But the migration over rescue, the isoform-specific effects on distinct cellular compartments, and the breadth of AKT's known substrate space all point to an AKT activity program that is considerably more extensive than the mTOR axis alone. What is that full program?
These are not merely academic questions. Karis Eng's work on Pten hamartoma tumor syndrome has established that Pten loss in humans produces a syndrome spanning cancer predisposition, epilepsy, macrocephaly, and autism simultaneously. Damon Page's conditional knockout models and Mustafa Sahin's circuit-level analyses have defined the behavioral and neurophysiological consequences. Antonio Hardin has pushed towards circuit-specific phenotypes, but therapeutic precision requires knowing not just that AKT is necessary, but specifically what it is activating and when.
Here is what makes the timing question urgent. Rapamycin can prevent these Pten loss phenotypes if administered early, but cannot reverse them once established. That developmental window means the AKT activity program is laying down changes that become permanent. Identifying the downstream effectors that AKT engages in newly born neurons before those changes become fixed is the difference between prevention and a therapy that always arrives too late.
What specific programs is AKT switching on in newly born neurons that drives their transformation into epilepsy-prone hypertrophic cells? Are there AKT substrates beyond mTOR that the field has been systematically overlooking? Do AKT1 and AKT3 activate distinct transcriptional or post-translational programs? And could that explain their differential effects on specific morphological compartments? Answering these questions will determine what a precision therapy for Pten-related epilepsy and autism actually looks like.
This paper changes how the field needs to think about the Pten signaling cascade. It is not sufficient to say mTOR is the effector, AKT is the necessary node, and the complete AKT activity program downstream of Pten loss remains to be mapped. That mapping will very likely determine the real therapeutic targets in Pten-related neurodevelopmental disease.
Definitive work from the Lucard and Weston labs, with Prina and Goyette as co-first authors. This is exactly the kind of paper that closes one major question and opens a field. Until next time.
Until next time.